For joining me again. Can you hear me okay?
I hear you okay.
Good. Now, you are in Europe and you believe in evolution, and we said you get to go first to define what you mean, and I'd like to hear your side of the story. Tell us, in case people haven't seen any of our previous debates, tell them a little bit about yourself, what kind of work you do, what kind of education, and I think define the word evolution for us.
Yeah, okay. First, my name is Stefan Frello. I live in Denmark. I'm a teacher for 17 to 19-year-old young people. I teach maths, and I have a PhD in botany and a master in molecular biology. To define evolution, well, first, what we agreed on was the title of our debate was can the observed variation between closely related species be used as evidence that all living species are related through common ancestors? And
Correct.
that is what I'm going to argue in favor of.
Okay.
To define evolution, I think I have, yes, I have it here. First, evolution is biological evolution. I know that some people use the word in various different other ways, but I'm going to concentrate on biological evolution, which also is clear from the title of the debate. And it works more or less like this, put up here. Any organism will produce more offspring than will survive, and those offspring will be genetically variable. Some genetic variations will influence survivability and reproducibility. And the genetics of those with better survival, or rather better reproducibility, will dominate the next generation. This would result in an accumulation of variation over the course of many generations. And of course, in order for this process to go on, new genetic variation has to be supplied by mutations, because otherwise, sooner or later, the variation will disappear, and only one single type of organism will be left. But if we can show that new genetic variation can be supplied, then the process can it should be able to go on forever. The consequences of this is that the accumulation of that there could be an accumulation of genetic complexity and of anatomical complexity as a consequence of the genetic complexity. And another consequence is that all living organisms are related through common ancestry. There are other mechanisms than natural selection, which was the one I described, and they influence evolution too, but natural selection is thought to be the only mechanism that can result in an accumulation of complexity. The other mechanisms is more a walk on the same spot, so to say, just adding variation, but not necessarily accumulating complexity. Now, natural selection do not necessarily result in an accumulation of complexity, but it will result in an ever better survivability and reproducibility of the selected organisms. So that is more or less the theory that I will defend. And just to make sure that we know where we agree and where we do not agree. You very often repeat this: dogs produce dogs, and all dogs had a common ancestor. It was a dog. And as a scientist, I would have to agree on that. All dogs produce dogs, and all dogs had a common ancestor, and it was a dog. There's no disagreement here. The disagreement comes in when we look at what happens on the long term. You often say we will never see a dog produce a non-dog. And within observable timeframes, I will have to accept and agree on that. Within observable timeframes, which is the normal lifespan of a human being or a few generations of humans, we will not expect a dog produce a non-dog. So actually, we do not disagree that much on this. It's not what can be directly observed we disagree on. It's more like what cannot be directly observed right now, but what can be inferred from the observations that we do. Because evolution, the theory of evolution says that from the observations we can do right here and now, we can infer that evolution actually has taken place and that genetic and anatomical complexity has actually accumulated. And if we go far back in time, then the ancestor of dogs were not dogs. And that is where the disagreement is, not on what we can directly observe. I also have to talk a little about what observation and evidence and confirmation actually is in scientific content, because direct observation of a phenomenon or a process is not necessarily a request as evidence. For instance, 350 years ago, it was well established that the Earth is orbiting the Sun, but nobody had observed it, and yet it was considered a fact. So that's just one example that direct observation is not considered a necessary request as evidence. And if we go just 100 years back, it was considered an established fact that matter is made of atoms, but at that time nobody had seen an atom. It is actually only in the recent time that there has been direct observations of atoms, and yet it was considered an established fact. Also, neither you or me or anyone else I know will dispute that fossils show that dinosaurs and numerous other extinct organisms lived on Earth. Nobody had seen a living dinosaur, and yet there's no doubt that dinosaurs were alive. So, just to stress it, it's not a necessary request as evidence that we make a direct observation of a phenomenon. We can infer it from indirect observations or of other observations that show that this process in question actually has taken place. Okay, so back to our topic. What should we expect to observe if the answer to our question is yes. And the question, just to make sure that everybody remember, can the observed variation between closely related species be used as evidence that all living species are related? See if I can hit it here. Yes. In general, the same kind of variation, we should see the same kind of variation, but not the same amount of variation at different taxonomical levels. We should find if we find a large amount of variation at the order or class level, I'll come back to that just in a second. If we see that, then we should expect small variations at the species or genus level. If we find large anatomical variation at the order or class level, then we should expect small variations on the species or genus level. And just to make sure that people are acquainted with the taxonomical levels, species could, for instance, be the wolf or the mallet, and representations of the genus. Canis lupus is the wolf, and Canis familiaris is the domesticated dog. The mallet and the teal is within the same genus, Anas. For the wolf is a member of the family Canidae, and so is the fox. The mallet is a member of the family Anatidae, and so is the goose. All carnivores, such as wolves and fox and cats, are members of the order Carnivora, and all ducks and geese and a few other birds. In this case, I have chosen the screamer, if anybody knows that. Is made of the same, is members of the same order called Anseriformes. And the highest level I have shown here is the class, very familiar, Mammalia and birds, called Aves in Latin. So what I'm talking about is that at the species and genus level, we should see the same kind of variation as we see at the order level, but at a much, much lower amount of variation. To make me a little more specific on what we should expect to observe, if the answer is yes, we should be able to identify mechanisms that can result in new genetic information. As you remember, if evolution had to be accepted as an ongoing process, there has to be some kind of mechanism that add new genetic information to the genomes of the organisms. Otherwise, we will soon run out of variation, and all organisms of a specific species will be identical or nearly identical. So some kind of new genetic information should be added to the genome, and we have to be able to identify such mechanisms. We also have to be able to see a change in fitness, and fitness is defined as reproductive success. Because if there's no change in fitness, it means that all the members of a species has the same reproductive success. And if all members of the species have the same reproductive success, then there's no selection and hence no evolution. So some indication of variation in fitness should be able to be identified. Also, we should be able to see small anatomical and genetic changes and all kinds of small anatomical and genetic changes at the species level within historical times or within observable time. Those are the three topics that I'm going to talk about. We should not expect to see larger anatomical or genetical changes, and larger could be thought of as family level, which you hopefully remember. The family level is for instance, the wolf and the fox, and higher level of that should be, for instance, the wolf and the cat. Variation like the differences between the wolf and the cat, we should not expect to see that at the short time spans, observable time spans. But of course, there should be some change. If everything stays the same within historical time, then we cannot expect evolution to work. So these are my suggestions to what we should expect to observe, which I will show that we actually do observe, and what we should not expect to observe. And it's very important to make sure that you understand that we shouldn't expect to observe just about anything. We should only expect to observe minor differences. It's not the reason I have written this hopeful monsters is because it is a widespread misunderstanding that evolution could go in giant leaps from one species to a completely different species in one or very few generations, but that is not part of the theory. Okay, I have put up in the upper right corner the three main topics that I'm going to talk about. Okay, I shouldn't talk too much, too long. We should have a dialogue here, so I would like you to comment what I have been going through. Especially, what do you think we should expect to observe or not observe? Is the answer is no to our question, which I guess that you would say that the answer would be. And also, what evidence, if any, would you accept as, or what observation, if any, would you accept as evidence for yes to the question.
All
Okay?
right. All right. Thank you, sir. Thank you, sir. Hey, Steve. Steve, I'm getting a ring back. I'm hearing myself twice, getting a ring back here.
Oh.
Okay. Stefan, are you hearing it twice or just me? Oh, there, I stopped. It stopped. Okay.
I only hear them.
Okay. It stopped, whatever you did. You had about 20 minutes by my rough estimation here, so I'd love to go back through your slides and ask a few questions. You mentioned we should not expect to see any changes in the short term. This can
only minor changes.
Yeah, right. I understand.
Minor changes.
So you have to infer. I guess then I'd like to know your definition of science. Is this part of science, or is this? I would put it in the category of religion, but maybe you have a different word. We don't see any changes. It's my definition of science is things that are observable, testable, demonstrable. Can you, if you can't see, but you can only infer these changes, can you demonstrate? What if you chose a creature that had a much shorter lifespan? There are bacteria that have a 20-minute generation time. Why don't we see it there? Because with a 20 or 30-minute generation time, we're really squeezing the effect hundreds of thousands of years into one human observation lifetime. So, but we don't see it there either. All we see is bacteria produce bacteria, always the same species. What exactly are you claiming then is, you're claiming time is going to help. And that's what I've said all along in my teaching for 30 years on this topic, that time is the hero to the evolutionist. Time can accomplish, in your mind at least, what we can't observe and see. This is why I've continually said evolution is a religion that people believe in, but it's not observable. So let's see, just a couple quotes you had here. I think you said it cannot be observed, it must be inferred. And then you said, if we go back in time, Stefan, we cannot go back in time. It cannot be done. You can infer, you can think, you can dream, you can hypothesize, but the fact is we cannot go back in time. So if we don't observe dogs producing non-dogs in the 5,000–6,000 years of human observation, what are you hoping 5 or 6 million or billion or trillion years is going to do? And you never mentioned what is the mechanism that's going to add new information. To change from, say, a bacteria to a dog would involve billions and billions of bits of additional information. Bacteria don't have hair, they don't have a nose, they don't have a mouth, they don't have ears. Where's this information coming from? I'd like to see your mechanism to add information. I don't think any evolutionist has ever been able to demonstrate that it happens. They can imagine that it happens, they can hope it happens, and obviously they believe it happened, but that is not science. That is religion. You mentioned dogmatically in one of your slides that no one has ever seen a living dinosaur. I don't think it's logical or fair for you to make such a statement, because I doubt you've talked to everyone that has ever lived in through history. There have been thousands of reported sightings in history of people seeing living dinosaurs, only they had a different name for them depending upon their country. I cover this for nearly 2 hours on my video number 3. Dinosaurs that have been seen all through history, only typically called dragons would be a generic term. Dinosaurs that are mentioned, ancient artwork, ancient carvings on stones, stories by the thousands. Certainly dinosaurs as dragons are mentioned in the Bible 34 times. There's Behemoth and Leviathan. God said Job had seen this creature. He said he was living with one. Job chapter 40, verse 15. Behold now Behemoth, which I have made with thee. He has a tail like a cedar tree. And then there's all kinds of evidence, I guess anecdotal evidence of hundreds of people who will swear they've seen live ones in the last 100 years. There have been dead carcasses wash up on the beach. And let me ask you this, Stefan, if somebody captured an apatosaurus, a 20-foot-long apatosaurus in the swamps of the jungles of Africa, and they put it in the zoo in Denmark, and you went there and looked at a living dinosaur, if somebody did capture one and put it there and let you come pet it and feed it and sit on it, would you then admit that the teaching of dinosaurs and man together is correct? Or would you then try to calculate some way how that one survived for 70 million years? And would you maintain your theory, your belief, I guess, that they died out 65 million years ago? I have been convinced for years that no amount of evidence, none, would convince a person such as yourself to change the teaching, to change the theory, the belief system that you have, because you really like it for some other reason, either because you get paid to teach it and that's a financial reason to continue that belief, or a moral reason, people don't like the idea of God telling them what to do, or a pride reason of they never want to admit they're wrong. But if one was captured and put in the zoo in Denmark, would you then believe in creation? Or would you then give up on the belief in evolution? So the key points I would like to make in my short time here, and I don't want to take 20 minutes, but you mentioned that it doesn't happen in observable time. It must be inferred. My point is, okay, great. That's not science. That's your religious belief. And then you said, if we go back in time, I would like to point out, Stefan, we cannot go back in time. You can't do that. So this, again, is a religious belief. I object to your statement that no one's ever seen a living dinosaur. And you said you're going to identify the mechanism to add new information. I would like to see that. This is, as far as my best count, my 150th debate. Congratulations. You're number 150. And I cannot find anybody who's ever been able to explain where added information comes from. How did the bacteria get the incredible amount of information to grow hair? Just getting hair to grow on anything requires something enormous amount of genetic information. The gene code, it would be thousands of lines of code in a computer necessary to cause this to happen, or to grow a heart or an intestine or reproductive organs or any number of things. We could go for hours and hours. But there would be billions and probably trillions and trillions of lines of code would have to be brand new lines of code in the DNA of the bacteria to change it from a bacteria to a dog. So I'll give you the time to answer these. I would like to know, where is the scientific evidence for these changes? Not only a mechanism. If you can dream up a mechanism, that's only step one. You now have to prove that it's actually happened rather than it's just imagination. So I would love to know, where is the mechanism for this to happen? How on earth could you prove no one's ever seen a dinosaur with all the anecdotal stories? And why would you think going back in time would help? We can't go back in time and go. All we've ever observed is dogs produce dogs. There are no exceptions. So I'm baffled, I guess, that you can continue to believe that it happened long ago and far away. We can't observe it today, even with something with a much shorter generational time, like bacteria. We don't observe it happening. All we observe is the changes that we do observe are usually detrimental or fatal to the organism. There certainly are none that I'm aware of that add new genetic information. So I do appreciate your faith and your great zeal for your belief, but this is not science. Okay, go ahead, Stefan.
Okay. Do you have my screen back?
I have your screen. Yes, your screen is up. Says the consequences of evolution.
Okay. Back to the evidence and confirmation. I think it is first on the dinosaur. If you could show me, or anyone could show me a living dinosaur, of course I would accept that it was a living dinosaur. But first, I don't accept all your stories. There are stories about mermaids also, centaurs, all kinds of different trend of animals, and all are stories. And if you look at how these dragons are described in different cultures, the Chinese version of a dragon is completely different from the European version of a dragon. So I don't think that's evidence of anything. And there are other, actually history is full of interpretations of observations that go beyond what is actually observed. as I mentioned, just two examples, that Newton 350 years ago was absolutely convinced that Earth was orbiting the Sun, though he had never observed it. I'm actually not sure that you can say that anyone has actually observed Earth orbiting the Sun even now. And atoms has been considered a fact for at least 100 years, perhaps somewhat longer, but only recently anyone had observed it. So this accept of phenomenon that you cannot directly observe is an integrated part of science. It was not a religion to Newton that Earth was orbiting the Sun. It was not a religion to Einstein that the matter was made of atoms. It has absolutely nothing to do with religion. And if you want to insist in that, I would like to hear you define religion, to give a definition of religion where evolution is religion, but the orbit of Earth around the Sun was not a religion 350 years ago. I would really like you to give a definition of religion where this is accepted.
Would you like to?
No, okay.
Would you like to stop and do these one point at a time? I think people are going to get lost.
Yeah, perhaps we could do that. Yeah, we could do that. Yeah.
Okay, I think you're purposely muddying the water by saying 350 years ago the Earth orbited the Sun. It was established in Galileo's mind or whoever's mind, and that no one's ever observed the Earth orbiting the Sun. There are different levels of proofs, I guess, for any number of things. Certainly in the field of science, there are different levels of proof. There seems to be overwhelming evidence that we can quickly interpret to say, yes, the Earth orbits the Sun rather than the Sun orbit the Earth. For instance, in order for the Sun to orbit the Earth, to go around once a day, the speed would be phenomenal at 93 million mile radius, pi times diameter. It'd be, I don't think it could move that fast. Whereas if the Earth simply spins, now you have the effect of seeing the Sun go around. So the Earth orbiting the Sun to take a year is very different than saying the Sun orbits the Earth in one day, which is what it would have to do. So I think we could do I think most people who've studied this topic would say it's pretty obvious that the Earth is orbiting the Sun. But you are right, no one's ever observed it. So that
and until 350 years, it was a well-established fact that Sun was orbiting the Earth.
That's an excellent point. The fact that something is a scientifically established fact in the textbooks or in society does not necessarily mean it's true. I would point out that the same thing is today. In your mind and in the mind of many people who have fields of science as their expertise, they think it's a scientific fact that evolution happens. Dogs came from an amoeba. They just don't understand how I can't see that. But you do believe dogs came from an amoeba if you go back long enough. I'll give you all the time you want. You can have trillions, quadrillions of years. Ultimately, to summarize it down into one sentence, I'm assuming I can speak for you and say dogs came from an amoeba-like creature, a single-celled creature. Is that what you believe?
Well, not an amoeba.
A single-cell creature.
I don't think that we should go into the details of the early evolution of animals, but it has actually been shown with some confidence that a certain group of unicellular eukaryotes are on the cellular level are almost identical to the cells of a sponge. They look almost the same. They have almost the same function. The basic difference is that in the sponge, all these different cells are grouped together in a single organism, and this unicellular organism is of course unicellular, though once in a while it also forms a colony of cells that works like a very, very simple kind of sponge. So actually that has been shown in some details how the very, very early evolution of animals has been going on. But never mind. Do you have any?
Well, I just want to I want to point out to the audience that you evaded the question. I said,
Didn't you ask me if we evolved from amoeba? And I answered.
Do you believe, just very simple, yes or no, do you believe animals, multicelled animals, like humans, do you believe humans evolve from a single-celled creature, whether it's an amoeba or not, a single-celled creature, in any amount of time? Yes or no? Do you believe that?
Yes. Yes.
Yes. Okay, good. And I point out, you are welcome to believe that. But that's not observable science. We observe amoebas and single-celled creatures multiplying all the time, in a very short generational time for most of them, and we never observe them producing anything else. So if you wish to...
Hey, can I show you?
Sure, please do.
Yeah. See, it's a bit down here. Here it is. Yeah, here it is. Yes, this one. Hey, what do we have here? We have two single-cellular organisms, one called Paramecium and one called Chlamydomonas. Chlamydomonas is the little green one with the two antennae, and the other one is the fluffy one. The fluffy one to the left is eating the little green one, but it can only eat it if it is unicellular. And it always is unicellular in nature. It has never been observed to form colonies in nature. But if you put these two together, then Paramecium starts eating the little green one, which is actually an algae. And as a response to that, for the first time in history, it forms colonies. And those colonies are actually a little different when you repeat the process, which I have illustrated to the right. The bluish, I don't know if you can see it in the middle, the B column. There is a kind of bluish shadow around some of the cells, and that's one way of producing a multicellular organism. That single cells are grouped together, and there's a common membrane or the mutual membrane around all of them, and then they release single cell individuals which form new colonies in the same way. Or the colonies can be constructed by the single cells kind of gluing together and then releasing single cells that grow in the same way and form colonies. So this is the very, very first step from a single cell organism towards a multicellular organism, that the cells do not leave each other when the cell division is finished. They kind of glue together and form a colony. And the next one is going a step further. This is...
let's go back to that one if we could. Go back to that slide. Are you saying that a colony of cells is equivalent to an organism like a dog or a human, which has obviously maybe 100 trillion cells in a human, that they're working as organs, not as colonies now? This is very different. I think they're confused about a colony getting together for protection or for food production or something. We have all kinds of farmers get together in one community and have a colony of people. We had the American colonies, but they didn't ever fuse to become one human. They're still individual humans, and these are still individual cells. The fact that you have a colony of cells cooperating in a common goal, protection from the paramecium in this case, is not evidence that they can become a single organism, like the humans are still producing humans, even though they have colonized together thousands of times. So I think you're very misleading in your illustration here. If you're misleading the audience to think that this is a step toward multicellular organisms, no, this is a multicellular colony. Please don't confuse the two. This is not evidence for evolution.
But what would the very first step from a unicellular organism to the multicellular organism? It had to be a colony of identical cells. That had
If
to
you,
be the first. It had to be the first step. What other step could be the first step, if
that's
not
a
this?
great point, Stefan. This is evidence that you have great faith that something did happen, and you're looking desperately for the steps of how it happened. But that's not
No, but
science.
I am looking right at the first step.
No, there are three. There are
Right
three
at the
things.
first step. And if you will allow me to show the next one, I am looking
Okay.
right at the next step.
Well, let me
Because
finish with this one. Let me finish with this one first. I don't want the audience to think that you're correct on this one. There are single-celled organisms, millions of them. There are
yeah,
single-celled organisms that work as colonies. That's
yeah.
very different. They're very different than a multicellular organism. You're assuming the colony is a step between the two. I completely disagree. This has not been demonstrated in any laboratory anywhere because you have to get a huge amount of new information. Each one of those cells that came into that colony had a gene code to make a cell just like itself, or with a few minor variations. None of these cells in this contain the genetic information to make hearts, livers, organs, skin, toenails, hair, teeth, eyeballs. Okay, and they're never going to get it. There's no new information in that colony. There's a new cooperation, but there's no new information, no new genetic information. So you're deceiving people to say that that would be a step to go from single cell to a multi-celled organism. The colony is totally separate, unrelated. That's a rabbit trail you're leading people down. Go ahead to step two, but I'm going to strongly disagree on your step one.
Okay, but if there is no new genetic information, why do they stick together? What
So
makes them stick together? What makes them stick together if not a change in the genetics?
was there a change in the genetics?
If the genetics is still exactly the same, then why do they stick together? Something changed.
It was in their genetics all along to stick together as needed.
No, it wasn't. No, Mr. Hovind, it wasn't. It has never been seen before. Never.
Has anybody seen new genetic information that tells these single cells to join together to a colony? There are people all over the world who are individualists, like Daniel Boone, go out across the wilderness by yourself. And yet most of the people want to get together into a colony, like the New England colonies that came over here from mostly from Europe. But they're still individual human beings. The human beings never grew together to be one 5,000-ton animal or creature. They're always individuals in a colony cooperating. All you've demonstrated is the cells can cooperate for a common goal so they don't get eaten by the paramecium. That's cooperation. That's not accumulation of new information.
But why do they cooperate if they still are identical to the original one cell organism?
Yeah, so they don't get eaten. Why do people colonize and get together so the enemy doesn't in a wartime? Why do they form armies? An army is not a single unit. It's not a one organism. It's a group of individual humans.
So what you are saying is that these single-celled algae has some kind of individual goal that we don't want to be eaten, so let's cling together.
Oh,
nonsense.
I have no idea.
You are constantly comparing them with humans.
I have no idea what they're thinking. But
they don't have a goal. They don't have a brain.
Right.
All they have is the genetics.
You have not demonstrated that there's any new genetic information, and you've not demonstrated that this is a step toward a multicellular organism like a dog or a human. That's a colony. Don't confuse people with trying to say this is similar. It's not similar. This is not a step. You have no link. Where are the two-celled organisms in anywhere in biology? You have a master's in biology and a PhD in botany. Where are the 2 or 3 or 4-celled? There are none. The closest we get is a colony of maybe 20 cells cooperating together for some reason. But again, each cell contains the same original genetic code. And I would say in a court of law, Your Honor, you cannot demonstrate that the individual cells don't already have something in their gene code to say, oh, if we're getting threatened, join together. Just like people, when they feel threatened, they go huddle together and make themselves an army or a colony or a kingdom. That's not evidence for evolution, Stefan. Stop deceiving people. Go ahead, step 2.
These single-celled organisms don't know that they are chosen, that they are going to be prey for this predator, not before they are eaten.
Does the single
They are swimming happily around in the water, and then suddenly, shoop, they are gone. They are not warned in any way. It's not that they can foresee that, oh, we are going to be eaten because this paramecium is swimming around here, so we have to do something. That's not what's happening. Either they are eaten or they are not eaten, and that's it.
Let's take one topic at a time. So, does the single-celled algae have an escape mechanism? Does it have a mechanism to sense danger? Why does it flee? When the paramecium comes to the algae, does it lay there and let it be eaten, or does it try to evade being eaten in any way? Are those two little hairs on the algae, are they actually a flagellated paramecium, flagellum that can spin, that allow it to swim away? I think you'll notice
I don't know if it has any of the paramecium.
Get a microscope that can view down to single-celled organisms. If an enemy comes in, almost all single-celled organisms have A, a method of detecting their enemy, and B, a method of trying to evade their enemy. None of them lay there and get eaten, or they would go extinct. All of them have some method of protection. So you're saying they just colonize for no reason. No, I think maybe they have a reason. What they think, I don't know if paramecium or these creatures think at all, but apparently they do have some kind of mechanism already built in the gene code to detect a predator or a danger and to evade it. I think it's been demonstrated thousands of times that some can even change their coating on the outside if a particular chemical is present in their environment. They will learn to protect themselves by developing a shield around them. This is not adding new information. This is a selection of information already existing in the gene code. So to go from a single cell to a colony is not going to help your theory. But I know you believe it and you want to believe it. But it's not science. This is your religion. I'll stick by that. Anyway, so you think the colonies is a step between single cell and multi-cell. I completely object. That is not correct. But go ahead. What's your step 2 going to be?
Well, first, I have to stress that this has been repeated several times, and at least some different mechanisms has turned up. The column B and C shows 2 different ways that these cells protect themselves. So this points rather to new genetic information than to some pre-existing mechanism. But this step 2, the natural step 2 in the course from a single-celled organism to a real multicellular organism would be first to form a colony and then to initiate some kind of cell differentiation. And the very first steps towards cell differentiation is illustrated here. I have to explain what's going on. They have selected for colonization by growing these fungi, it is, in water that is not shaken, so that the cells would settle, and colonies of cells would settle faster than single cells. And by sucking up the cells that were in the bottom of this this tube, they got an enrichment of colonies of cells. And when they observed them, they observed something quite extraordinary, because they actually observed cell differentiation. They were able to stain these cells in various ways, and by doing that, they could see that the cells were actually behaving differently. It is illustrated here by some of them being blue and some of them being green. And it turned out that the cells that were different from the majority of cells actually were those that later died and caused the colony to separate into several colonies. So here we have the first step is colonization from single cells to colonies. The next step would be the very first initial steps towards a cell differentiation. And what we see here is actually cell differentiation. Some cells die, which helps the colony of cells to divide into separate colonies.
All right.
And this is not just a second, and this is actually beneficial for the cell colony because a large colony of cells would the nutritional supply to the cells in the middle of the colony would be less efficient than if the cell colony divide into separate colonies and then keep on growing. And that is what we see. And what the major point here is that they could identify the cells that later would cause the colony to separate into separate colonies. They could identify them several cell generations before it actually happened.
Okay, let me give my answer to that. Obviously, if there's a cluster of cells that have colonized, the ones in the center are going to get less nutrients, we would assume, because they have less contact with their environment. They're surrounded by other cells and they can't eat, sleep, breathe, or poop. So, of course, that's the place where the colony is going to separate. Those are the cells that are going to die and no longer cling to other cells. But again, I would point out that cells getting together and colonizing and then even differentiating, whereas different ones have different functions in the colony. In the American colonies, people came over, they settled, and very soon after they got the basic needs met, some became farmers, some became bankers, some became cooks. They had different activities, different specialties that they would, but they're still individual humans. They never merged into one 10,000-ton human being. They were each individual people in a colony. All you've demonstrated here is the same thing. A group of cells can get together, and maybe one is better at taking in a certain substance that the colony needs for survival. One may be better for defense, but they're still individuals, and I would point out that you've not demonstrated that this could not already be in the gene code. There are individuals who can live completely on their own, completely. They can go out in the woods and survive on roots and berries and catch animals, but if they join in with a group of other people, pretty soon they take on their specialty of maybe they're the marksman shooting game or the gardener raising crops. They're capable of living on their own, but they're also capable of living together with others. What you've demonstrated here is single-cell organisms are capable of living on their own, or they're capable of living in a colony. And if they get too big of a colony, the ones in the middle are going to die and hence break the colony at that point. So yes, I would think that's probably scientific and logical that you could look at a colony and say, that one's going to die and this is where it's going to split. This is not evolution. This is no help for your theory whatsoever.
Oh yes, because they could identify the cells long before the colony actually grows so big that that the cells were in any problem of nutrient supply. But I think we should leave that. We will not agree on this. So we can just agree on disagreement. Okay. I have presented the evidence, and you have made your comments. Back to the mechanisms for adding genetic information. You were asking about mechanisms for additional genetic information, and I just have to see if this will follow. Yeah. Just have to point out the right place. Here we are. Creationists often claim that no known mechanism can add new genetic information, but if you look at the scientific literature, there are actually several known mechanisms that can add new genetic information to an existing genome, and I'm going through a few of them. Within the last 2 decades or so, it has appeared to scientists that actually there is brand new genes called de novo genes or orphan genes, which appears from non-coding DNA. So you have a protein coding gene where no protein coding gene was in the ancestors of this.
Okay, hold it. Let's take it. I want to take these one at a time so nobody gets confused. Are you saying there were new genes that came from non-coding DNA? Was this new information added to the DNA or silent genes that were activated that were already present? Are you saying there was new information added, or just something that was normally non-coding all of a sudden started doing its job? Someone came off of welfare, in other words.
I think I have to explain how these were identified. I think that would answer your question, if I may.
Okay.
Go through that. Just to make sure that that our audience are familiar with what a gene and what a protein coding gene is and how it works. DNA is built of nucleotides. It's this well-known double helix structure, and all the little balls in the middle there are representing the so-called nucleotides. There are 4 different nucleotides called, normally abbreviated A, T, C, and G.
Right.
And the function of a protein coding gene is that the DNA is transcribed into a so-called messenger RNA molecule, and that molecule is translated into a protein. It's a complicated mechanism, of course, but that's a short story of it. And it is the protein that has the function. The gene itself, the DNA gene, doesn't have any other function than being transcribed and translated into the protein, and it's a protein that actually has a function. This is going on by taking 3 nucleotides at the time and translating it into 1 amino acid, and a sequence of amino acids then builds the protein. So it is the sequence of amino acids that decides decides what the protein will be doing. We'll skip this one. Then identification of candidate new genes. This is, I think, what you were asking. How do we identify a new gene?
No, I'm asking, not is it a new gene? Is there new information added, or is it a scrambling of existing information, or a gene that came off of welfare and decided to get to work now?
Yeah,
it's already there.
This is more or less the same. What people have been doing for identification of these new genes, they have compared a number of very closely related species, and often it's species within the same genus. Often they have been using Drosophila, coming right up there. Drosophila is a fruit fly.
Right.
And there are a large number of, a very, very large number of Drosophila species, and I think that we can agree that they had a common ancestor, which was a Drosophila fly.
Fly, yes.
Just to make sure that we know what we agree on. And then a number of these different Drosophila species have been analyzed in very, very great detail. The most well-known one is Melanogaster. That's the one that has been used for genetic research for decades, or almost 100 years now. And in Melanogaster and its closest relatives, or the species closest related to it, researchers has found a number of protein coding genes that are not present in any other Drosophila species. And there's a very, very big number of Drosophila species that has been investigated here. So the simplest explanation is that these genes actually occurred in the common ancestor of Melanogaster and its very, very close related species, and that it is not a gene that has been lost in any of the other species. It has never been there. It has been the sequence that make up this gene is present in the other species, but it is not an active gene, and it has never been an active gene. It's not a sleeping gene that just waits to be activated. It's a sequence that is not that doesn't have the right signals for being translated into a protein. Actually, what they did in this particular investigation here was that they were able to identify the so-called stop codon. If I go back a few slides here, a protein coding DNA sequence is defined by its start codon, which always is ATG, the start codon, and then a number of amino acid defining codons, and then a stop codon. And between those two is the protein, to tell the story very short. And what they were able to do in this investigation, in this slide, come on, here, and no, this one, this one, and this one. Those two are both Drosophila investigations. And what they were able to do is to identify the mutation that removed a stop codon in the middle of this DNA sequence, so that instead of a stop codon, you have an amino acid defining codon, and then what was a non-coding DNA sequence suddenly was turned into a coding DNA sequence, and now the protein was able to be translated and function. So if that is not new genetic information, I don't know what you are asking for.
Okay.
This is a protein that is produced in some species that has never been produced anywhere before. They have tried to search the entire gene bank, which comprises millions and millions of DNA sequences from hundreds of thousands of different, perhaps not hundreds of thousands, but tens of thousands of different species. And they haven't found one single example of this particular protein in anywhere. So it is a new protein.
Well, it's a new protein, possibly. Let's discuss this for the listeners here. What the stop codons would be similar to a period when you're writing the English language, and I'm assuming it's the same in Danish or Dutch or German or French. You have a sentence, a single thought, and you put a period at the end, and now you start a new thought. If by accident one of the periods was taken out, you would put two sentences together and get in English what we call a run-on sentence, which now may give a little slightly scrambled information to the person trying to read it, like what does this mean? So I think what you've done is you've discovered that in a certain few cases with the Drosophila fly, the stop codon, the periods were removed, and it developed a run-on sentence. And they're calling that a new protein. Okay, let's call that a new.
They're not calling that a new protein. It is a new protein.
It is. It's a new protein.
You have to understand this. Before this stop codon was removed, there was no protein. No protein was produced. Right. Only after the stop codon was removed, a protein was produced.
I would like to point out that this is not new information. The information was already there. It is now scrambled into a run-on sentence. And I'd like to point out, it is still a fly. In every case, it's I mean, are you telling me there's been examples where it turned into something that was non-fly?
May I remind you, Mr. Hovind, that at the very beginning of this, I said that, yes.
Have to wait longer. More time.
In observable time frame, we will not expect to see a dog produce a non-dog. That was what I said, and you insist that that is what we should expect to see. Actually, you insist that in order for you to accept evolution, we should observe what the theory of evolution says that we should not observe. That is nonsense.
Okay, I am pointing out for the listening audience here that what you've given us is an example of removing a stop codon, creating a new protein, which I will give the analogy of a run-on sentence, which would be a whole new thought. Two sentences run together, like, whoa, it's a new sentence. I agree. It's not new information.
The information before that new sentence was not two sentences. It was 2 times nonsense.
Right. So that is not a mechanism that's going to change a single-celled organism to a human. This is not observed, and you fall back on your God, which is time. Time can somehow solve it. This was a loss of information. Have any of the flies that have been observed to have this new protein, first of all, have any of them been an improvement over the fly? Secondly, who are they going to marry? Or is this new protein going to be lost in the next generation? Because you'd have to have two, a male and a female, in the same place at the same time and be interested in each other. You've got a host of problems. If this happens once by random chance in one fly gene code, how's this going to get passed on? Has it ever been demonstrated that it gets passed on? And is it new? Is it helpful to the organism or harmful? They've got
Yes,
flies that are blind, flies that can't fly, flies with curled wings. They don't get an improved fly.
well, actually,
You believe it. You believe it.
some of these identified new genes actually has been proven to be essential to the fly. Probably because it has lost some other gene and this new gene has replaced it, or because some other function has developed in or had evolved in the fly so that it became dependent on these genes. So actually, some of these genes are now essential to the species where they are found. So yes, it is actually new information, and the fly has changed. The new species. It's not a non-fly, which you should not expect it to be. It's a new species, which is what you should expect it to be. And just to make a little, well, what do we see here? There's a number of pictures. What do we see?
I see a variety of dogs, but let me point out.
No, you don't. Mr. Hovind, no, you don't.
Oh, okay. There's a panda bear in there. There's a panda bear.
No, it's not. No, it's not a panda. That's a dog. But the one the one to the left is a cat.
Oh, okay. But Stefan,
apparently, Mr. Hovind, apparently cats and dogs has an overlapping variation.
Whoa, they both have two eyes, and they're both affected by gravity. That proves it. Stefan, hold it. We started late because of the technical problems, and I apologize. I'll do this again anytime. I have another debate, so we have to stop in about 15 minutes, and then I have to stop. I have another debate already scheduled. So yes, good. You got a cat with long hair that I mistook. I just glanced at him, mistook it for a dog. That's, I think, evidence that the same designer made them. Some people confuse the Porsche and a Mercedes. You know, when they go by at 200 miles an hour. Well, what was that? Well, they both have 4 tires. They got to be related. No, it's the same. It's a common design, and I don't think I'll ever get you to see that. But I want to go back to your fly illustration. You're getting a new protein from this stop codon being removed. It is still a fly. You still have a problem of this being passed on to the next generation. And where is the example of an improvement of changing to anything other than fly? This isn't help for your theory.
We shouldn't see anything other than a fly within historical time.
So because we don't see evolution happening, that's proof that it happened.
No, we see evolution happening. We see new genetic, new proteins, new protein coding genes appearing at these flies, and we have seen them all over the place. It has been. This is another example of Drosophila. This is humans. This is mouse and rat, and this is fungi and mollusks and tardigrades and nematodes and plants. It has been found all over the place. And Mr. Hovind, this is actually the mechanism that you were asking for, because if within a time frame that is short enough for an organism still being in the same genus, like the Drosophila here. Drosophila is what we normally call fruit flies.
Right.
Within Drosophila, we have several examples of new genes. 195 new genes has been identified in various Drosophila species. Just think for a second. If 195 new genes can emerge within a single genus, then how many thousands can emerge within a family or within an order? And that is what you are asking for. New genes that change the genome of the organism, and we see it right in front of us.
Hold it. You're moving the goalpost here. Let me ask you this. I'm just maybe totally unable to give you information. How many genes are there in the fly's gene code?
About 20,000.
20,000. So
yes,
you're saying
this is actually 1%. This is actually about 1% of it.
So out of 20,000, you're saying 1% of the genes have been modified through losing a stop codon, Again, they're losing information. They're not gaining information.
That's not. That's nonsense. Losing a stop codon is not losing information. It's changing one nucleotide into another nucleotide so that a stop codon turns into an amino acid codon. It's not losing information.
And are you convinced that that mechanism you just described of losing a stop codon and creating a new protein, that is the mechanism that is going to, over trillions of years, going to change a single-celled organism to a human and an elephant and a whale? Is that what you believe? This is the mechanism that will do it?
Plus a number of other mechanisms.
What other mechanism?
Well, we don't have the time for it, but I have a number of slides showing other mechanisms too.
Go ahead.
There are plenty to take up, but if you think about it, 1% within one group of organisms that are so closely related that even you would accept that they had a common ancestor.
Right.
So if you just for a short second accept that this could be extrapolated to family order, then you would not have 1%, you would have 10%, 50%. You would have almost all those genes that are not essential for the very basic biochemistry of the animals. Okay.
Let me give an example. When they first started the Olympics, I think 1930s or something, Hitler revived the Olympics idea. We could look at the top speed of people running, and let's say someone could run the 100-yard dash in 10.5 seconds. Over the last 70 years, they've got it from 10.5 down to under 9 seconds now, I believe. If in 70 years we can knock off 1.5 seconds, does that mean if we go 700 years, they can knock off 10 seconds and go down to 0 and run the 100-yard dash in 0 seconds? Is this logical, or is this stop codon, is this new, this protein mechanism that you just described, could it be that that has a limit? And you got 195, maybe they're only going to get up to 250 and something's going to shut it off, or the fly is going to die. You haven't demonstrated, other than imagination, SpongeBob, that maybe if we give it more time, there's your God, it could do it. This isn't science to say that. You can't go back in time. We can't go forward in time. All we've observed in all of human history is flies produce flies. If you want to say they have new information, did the fly get something different than a wing, or did it get a new structure? It had a new protein. What good did this do for the fly? Can it fly faster? Can it produce more offspring? What beneficial mutation has come from these 195 new genes in the fly out of the 20,000? Have you demonstrated any new information that's helpful to the fly?
Well, as I said, a few of them have actually turned out to be essential for the survival of the fly. I don't know if they have been analyzed so that people know what they actually do, but they have been demonstrated to be essential for the survival of the fly. So in some way they are very, very important, some of these.
Well, now hold it. Are they essential to the survival of the fly because it is now encountering some new environment, like some man-made toxin or poison, or something in the atmosphere or the food? The world is constantly changing, sometimes man-made, sometimes nature-made. But did the fly ever get a turbocharger? Did he ever get a jet engine, or is it still a fly?
As we should expect under the theory of evolution, it is still a fly, yes.
Hold it. As we should expect under the theory of creation, it is still a fly. Creation predicts that also, by the way, Stefan. Creation predicts animals will always bring forth after their kind, and a fly is the same kind of animal as a fly. So this is predicted by the creation model, not by evolution.
I would love to discuss phylogenetics with you and show you why phylogenetics is not, as you describe it, just lines on a piece of paper, but that it is actually very, very well founded in observation, because that would be what you are asking for. How do we know that different animals are actually related? What are the evidence that organisms or animals that you would say were different kinds, even though you have never been able to demonstrate that you actually know what a kind is. On the contrary, you have been contradicting yourself all the time on that, showing that well, I have to be blunt with you. You have been showing that you have no idea what you are talking about when you mention the word kind.
Okay,
but,
and let's go on.
back to the question of this.
You have no idea what you're talking about when you mention the word evolution. You have no idea what you're mentioning about talking about when you talk about animals producing new information. That was not new information. It already had the sequence of letters. It removed a stop codon. It happened to make a new protein, but the information was already there. You didn't add anything to the gene code. You have no clue what you're talking about. Now, we only have 6 minutes left. Can you give a summary of your position? We'll do another debate as often as you would like. We'll do this every day if you'd like, but I ran out of time. So give me, you said at the beginning, and I pointed out, that you said, we don't observe it over the short term, and we should not expect to observe it over the short term. Okay, so therefore, my point is, this is something you believe in. You don't like the word believe, but that is very much what you believe in. So do you have one more point you'd like to make, or maybe a closing, or draw your pieces together? You're saying removing stop codons and creating new proteins in Drosophila fruit fly is enough evidence to make you believe that flies are related to bananas and elephants.
No. Well,
okay.
If I was going to present all the different arguments in favor of evolution, it would be a 2-hour non-stop speech or something like that.
Okay.
If people are well, you really have to. I don't know what to do for comprising the evidence into a reasonable time. But I think we should conclude this, because you have to.
Yes. Yeah. Please do. Go ahead. Give a closing. Give a closing here, and we'll take up round 3 later.
Yeah. Okay. First, I have to stress that that it is not a necessary request of evidence that a phenomenon is directly observed. It is sufficient that you have that you can say, well, what we see is what we should expect to see if this phenomenon was actually occurring. But the phenomenon itself is occurring over a very large amount of time, so we cannot expect to see it. But if this phenomenon, evolution, occurs over these large spans of time, not trillions of years, as you said, but up to, I think it's about 600 million years is the time frame for animal evolution, or perhaps a little more. And if animal evolution has been taking place for 600 million years or a bit more, then what would we expect to see? And one thing we would expect to see is new genes going into the genome of organisms by whatever mechanism can make new genes arrive. And that is what I chose to show you here, that process has actually been established. Several other processes for adding new information to the genome has been established too, and I would love to continue this and show you. But what we have to appreciate that the phenomenon does not have to be directly observed in order to be a scientific idea, or even to have to be a scientific, well-established theory. And that would be my concluding remark.
Okay, very good. Thank you, sir. And let's do another one here. What would we expect to see if God created basic kinds of animals, gave them a phenomenal gene code, the ability to produce a wide variety within that same code, big dogs, little dogs, long hair, long legs, short legs, but always a dog? What would we expect to see? I think we would expect to see bacteria produce bacteria, flies produce flies, but dogs produce dogs. I think that's not only what we would expect to see, that's what every farmer on planet Earth for all of human history will record. That's all they have ever seen. So not only do we have, I think, the common sense expectation on the side of creation, We have all of observation. All human observation says animals were created to bring forth after their kind. Any scrambling of the information usually results in a mutant, which is harmful or fatal. Many of them, of course, cannot reproduce, and you have to have, if you're going to get a beneficial one, which has never been demonstrated, you'd have to get two at the same time of the opposite sex in the same place, and they now would have to not only take over the population, all the rest have to die, or it doesn't work. Evolution is a religion of death. Everybody has to die who's not fitter. This is Adolf Hitler, 101. These are the unfit. Let's kill them off. The fit need to survive and take over the population. You folks in Europe are very familiar with that. So you say it does not have to be observed. I point out, Stefan, it isn't observed, and you believe it, and I appreciate your faith, but it's not science. And thank you for coming. Let's do another one. If you can call my secretary, I do have to go. But do you want to give folks your contact info if they want to get a hold of you? Stefan, do you want to give people your contact info?
Yes, it is stfr at hotmail.com is my email. Feel free to write any question to me, and this slideshow I will put it on my homepage. The address of that was on my very first slide. This one here.
Yeah, call that up.
Evolution and some strange number.
There it is.
It will be there in a few days.
creationviaevolution.dk/43647744-2. Okay. Well, thank you so much, Stefan. Let's do another one. And I'd like to see what other evidence you have for evolution. Appreciate you coming on. Thank you.
Thanks. Bye.